Hello everyone. So if you remember, we had this specific video tutorial uploaded earlier where we talked about loading the single cell sequencing data using the Python based program, specifically ScanP. And for example, you received the first few files from your run in the machine and then used sort of star based method. For example, cell ranger or something like that to generate your matrix files. Now you want to analyze those files and interpret your own data. To do so, you could use Python based programs as well as R based programs. And for that analysis, the first and most important step is loading your data. So to load your data, as I have shown earlier on this video how you can use the Python based program to load this data. Today I plan to demonstrate how to load this data for further analysis using R based programs. So very first condition is you need to install the R, then RStudio. And R is quite famous for extensive statistical analysis and an amazing platform for visualization of your data. So if we think at the very first that what are the currently available programs, those in R we can use for single cell sequencing data analysis. It could be single cell RNA, single cell protein, or combination of both of them, site-seq as well as single cell heterogeneity. We can use single cell DNA for different purpose, but I'm not going to talk about that today. So the next step in here is finding that program is a challenge. Rahul Satija lab from NYU have written this specific program called Seurat. Seurat is solely one of the most popular program which is used for single cell data analysis. The version 5 of it is already in the GitHub, but the crane, within the crane where our packages are installed or deposited actually for installation in RStudio, they'll be submitting it day after tomorrow, and then if crane accept, then you will be available to directly install it. Now let's go and open RStudio. So first we need to install the RStudio. To do so, you can just go to the RStudio and install it. The first phase in here is installing the Seurat package. So install and Seurat. Enter, it will be installed in here. The next phase, when it's installing, let's look at the data formats we have. When we generate by select gene or something, the data are in multiple formats. They could be in matrices format like barcodes, genes and matrix. They could be compiled or compressed as .h5 format. These are the most two common and popular formats that people use, and the data is actually generated after FAST2 processing. And when you process your data in RStudio and after you save it and you want to reload and you have to do the further analyzing, that .rds format is in there. If you are extracting any data in there, you could get it as a .csv format. If you are downloading some data from the geo, it could be something like this .txt. So we will go through each of these and try to load all these data formats for further analysis. So hopefully the Seurat is installed. So we do delete it and let's start the section. At the very first, let's say our data is in this matrices format. For example, barcodes, genes and matrix format. I want to load this data file. So we copy the path, and in the meantime, let's start writing. So we name it like Seurat _ mtx read 10x. There's nothing coming. So what we do, let's import the library first. Library Seurat. Let's see how it goes now. So we have imported the Seurat package, and let's do the same thing again. Seurat _mtx. Now when you write the read, you will automatically see that read 10x is coming, which is specifically say that your sparse matrices data you can load by using this read 10x. So we wrote it. Now we want to detect actually show the directory or where this data is installed. So this is the link where we have our data and we want to see. We run it, and what we see, we have successfully loaded the matrices format. Now the second step for it, that now if we can easily on this way load our data which is in the format of matrices as a sparse matrix. Now we think that okay, our data is compressed and it came as .h5 format and we need to load it, and to do so, first thing we do, we write Seurat _h5 and then start reading it. See, the H5 format is in here. So, one thing that I actually like to show in here, let me show. If we click on this read H5, it comes as a format of read 10x HDF5 file. So, HDF format sometimes is not installed automatically on your system. If you are using Linux based system, probably it's not installed. So, you need to use the command line to install it separately. So, that is a different perspective, and you can load the data from single cell ATAC-seq as well as single cell RNA-seq in this format. So, now we have it. Let us give it a name, file name, and then show the path where our data is, and we want to see that if it is loaded or not. Let us run it. So, as you can see in here, it is already loading our data set, and it successfully loaded. Now, these are the most common format where the data is come. Now, if you think that you got a data set which is actually not on these two format, you downloaded it from geo and data is in .txt format. So, before that I would like to add up one point actually, that if you are processing using cell ranger, it will also produce the loop files. So, if you want to see or visualize your data, one of the major option is you can use the loop browser from the 10x. That will be help you to visualize your maps. So, that is one of the option. But either way, let me go through this. If you are using now if you download from geo or somewhere, data formatted .txt. So, you want this to be loaded in your RStudio for, and you want to make it a, I would say, Seuratobject. So, what do you do? Data, and then this data is in table format. So, you read it as a table, and then we put our data. Now, we want to make it a Seuratobject. So, to do so, let's say we have the data table or text file Seurat, and we name it as create Seuratobject, as you can see in here, and then we go for the counts and we go for geo data, and then we want to see what we have loaded and how is our Seuratobject. You can see let's load it and convert it as a Seuratobject. Now, for example, you are analyzing, and in the middle you have extracted some data as a .csv format, and you want to load it. And to load it, what you have to do, you want to load it in this RStudio, and to do so, let's load the CSV format, and we use the inbuilt function of read CSV. Sorry, I probably put one extra, and then we want to see it. So, this is the data for CSV file, and you have all the parameters those you are interested, like you have base, mean, and everything you are interested in there. Now, you did all your analysis and you see your subject or analysis the Seuratobject as a .rds file like this one. You want to reload it and do the further visualization or analysis. And to do so, we can use the specifically Seurat function. So let's say Seurat underscore object file, and we lead it read RDS. So this is the function to write the single R object file and restore it. So we write it like this. We name it as a file, and we copy the link. And this is loaded. So these are the multiple formats of your data and how you can load them using specifically the RStudio. It's single cell sequencing data and go for further analysis. On the next verse of the video tutorial, I'll be uploading as using the Python based program and R based programs the demonstration of the pre-processing of single cell data. We'll begin with single cell RNA-seq. We'll eventually go for elaborate details. These are the way we need to do it. If you need any sort of help or with any of these part, feel free to ask me and anything you require. Thank you and see you.